on to the next speaker, Dr. Claudia Barth. She is a so she couldn't make it here today in person, unfortunately, but she sent us a pre-recorded version of her talk. And she is a senior researcher and PI at the FemHealth project in Oslo in Norway, and she will be covering aging, focusing on female aging.
Hi everyone, my name is Dr. Claudia Barth, and I'm a senior researcher based in Oslo, Norway at Diakonhjemmet Hospital. Today I will be talking about how we leverage big data and causal approaches to study female brain and mental health and aging, but first a little bit of background. I have no disclosures. As you all know, there are prominent sex differences in the risk of a variety of affective and neurodegenerative disorders. For example, the risk of developing depression and Alzheimer's disease is 2 times higher in females than in males. However, this disease risk is not stable, but changes across the lifespan and parallel to hormonal transitions. Here in red, estradiol, the most abundant and potent estrogen in the female body, fluctuates over the course of each menstrual cycle. When pregnancies occur, it rapidly increases during pregnancy and declines postpartum, and it further declines during the transition to menopause. These major shifts in estradiol can predispose females to mood disturbances with a heightened risk for developing clinical depression, such as premenstrual dysphoric disorder in the premenstrual phase of the menstrual cycle, peripartum depression, most prominently postpartum depression after giving birth, and perimenopausal depression during the transition to menopause. While estradiol seems to protect females from cognitive decline during reproductive years, low levels of estradiol after menopause have been associated with females' increased risk for Alzheimer's disease in older age. Precisely how estradiol and female-specific reproductive factors known to impact estradiol exposure affect females' vulnerability for affective and neurodegenerative disorders remains a matter of debate, as it's not easy to study, in humans particularly. So the question arises how to best study the impact of factors related to estradiol exposure on female brain health with relevance to depression and Alzheimer's disease. I would argue that we need 3 key research pillars. First, we need granularity to allow for comprehensive phenotyping. Second, we need scale for big data mining to increase power, reproducibility, and generalizability. And lastly, we need state-of-the-art methods such as machine learning. While we have employed all 3 components in our research, today I will highlight how my research team used scale and methods in the pursuit to study brain and mental health in females. For scale, we used data from the UK Biobank. The UK Biobank is a large-scale population-based health study from the United Kingdom that includes genetic, medical, lifestyle, and imaging data from around 500,000 participants aged 40 to 70 years at baseline. Using this data set, we asked the question how female-specific reproductive factors such as a history of childbirth, reproductive lifespan, and the use of exogenous hormones in form of hormonal contraception and hormone replacement therapy impact the aging female brain. As a proxy of brain health and aging, we calculated the so-called brain age gap. The brain age gap refers to the difference between a person's estimated brain age calculated from magnetic resonance imaging data using machine learning and their actual chronological age. For example, if someone's chronological age is 50 years, as depicted here in this figure, and their predicted brain age is 45, the brain age gap is minus 5 years. A negative brain age gap indicates a so-called younger-looking brain relative to a norm population, while a positive brain age gap suggests a brain that appears to be older. Using brain age gap as a proxy for brain aging and health, we found widespread associations with female-specific reproductive factors. For instance, we found that a history of childbirth was associated with a younger brain age, even decades after individuals have last given birth. And these effects were most prominent in the basal ganglia and the limbic structures. Interestingly, we also found a younger brain age in fathers, but the effect in basal ganglia and limbic structures seem to be specific to the mothers or the females. Second, we also found that a longer reproductive lifespan with a later age at menopause was also linked to a younger brain age, and we found this association both in cross-sectional and longitudinal studies using UK Biobank data. When it comes to hormonal contraceptive use, we actually found no association with brain age. Interestingly, looking at cognitive data in the UK Biobank, we actually found quite pronounced effects of hormonal contraception and cognitive performance later in life with mostly higher cognitive scores. So there seems to be this discrepancy of no effects on the brain, but on cognition in the UK Biobank. When it comes to hormone replacement therapy, we actually found an association with an older brain age. However, the effects were kind of at max 9 months, or would translate to a difference of 9 months. So I could argue, or I think we all could argue, that that might not be clinically relevant. Furthermore, also the effects appear to depend on genetic factors and a history of gynecological surgeries. In detail, we found that starting HRT earlier, particularly before menopause, was associated with a younger brain age, but only in females with an ApoE4 allele, which is a known genetic risk allele for Alzheimer's disease. Similarly, having high estradiol levels during menopause was associated with an older brain age in ApoE4 carriers, but not in non-carriers where the effects were reversed. Furthermore, we found that HRT use or HRT users with a hysterectomy, so the removal of the uterus, and or bilateral ovarectomy, so the removal of both ovaries, had a younger brain age than HRT users without such surgical history. This finding was particularly interesting as previous studies linked such surgeries to adverse effects for brain health. However, these studies didn't compare to other HRT users without surgeries who might be using HRT to minimize menopausal symptoms such as hot flashes and night sweats, which are neurological in nature. So the need for HRT could arguably be an indicator of neurological changes in women with menopausal symptoms. So for our study results, it could be possible that the timing between HRT use and surgery is much more tightly controlled than it is in women using HRT with menopausal symptoms, and therefore we find more beneficial effects for brain aging. But again, the effect size is also very small, so most likely not clinically relevant. But really more research is needed to establish a clear link here. So in summary, we found evidence that female-specific reproductive factors known to impact individuals' estradiol exposure, such as a history of childbirth and age at menopause, may influence brain aging later in life. However, due to the cross-sectional and observational framework we have been employing in these studies, we do not know whether any of these associations might point towards causal links. To move away from a purely observational to a causal framework, we ventured into the realm of genetics, applying Mendelian randomization, which I from now on abbreviate as MR, to test for causal effects of estradiol and female-specific reproductive factors on brain and mental health. MR is a statistical approach that uses genetic variants to probe the association between a genetically predicted exposure, for instance estradiol levels, and an outcome, for instance brain age. As the combination of genetic variants a person receives at conception is presumably random, MR is the natural version of a randomized controlled trial which is less prone than observational studies to confounding and reverse causality. My PhD candidate Hannah Oppenheimer implemented MR as a form of instrumental variable analysis, where genetic variants, here single nucleotide polymorphisms, SNPs, derived from genome-wide association studies, GWAS studies, are used as proxies, instruments, for the exposure and outcome of interest. Generally, MR operates based on three key assumptions. First, the genetic instruments are independent of any potential confounders that impact exposure and outcome. Second, the exposure is directly associated with genetic instruments. And third, genetic instruments affect the outcome only through the exposure, so no pleiotropy. But to be fair, that's the hardest to ascertain. More specifically, Hannah used a two-sample MR design, meaning that genetic associations with the exposure and with the outcome are obtained from two independent samples. Using the setup, we first studied whether genetically predicted estradiol levels are causally linked to brain age, depression, and Alzheimer's disease. To enable such an analysis, Hannah ran and annotated the first genome-wide association study of brain age in females. However, using this approach, estradiol was not causally linked to any of the outcomes. This finding was replicated using a male-only sample, using a multivariable MR approach accounting for body mass index. It was also replicated stratifying samples by menopausal status, so in pre- and post-menopausal women. And lastly, it was replicated using an independent sample, such as the life cohort from Germany. Next, we tested whether female-specific reproductive factors, such as reproductive span, age at menopause, age at menarche, and number of childbirths, all factors which were associated with brain age in observational studies, were causally linked to the outcomes of interest. The short answer is they were not. Lastly, we tested whether exogenous hormone use and gynecological surgeries such as hysterectomy and bilateral ovarectomy, also factors which showed up in our observational studies, were causally linked to the outcomes. Again, they were not. However, here we need to acknowledge that these exposures are lifestyle choices, or can be discussed as lifestyle choices, and health-related procedures which might not have a very strong genetic component. So, in summary, our work suggests associations between reproductive factors and hormone use in brain aging in observational studies. Yet, we found no causal links between estradiol levels and female reproductive factors in brain age, depression, and Alzheimer's disease using MR. This lack of causal links might be because MR inherently captures constant effects of genetic variants linked to an exposure such as estradiol. It doesn't capture estradiol fluctuations across the female lifespan, as I have shown to you in the beginning. So we could argue that dynamic effects of estradiol matter for female brain and mental health, and not constant or overall lifetime exposure. However, there are numerous challenges in studying these associations. For example, in the UK Biobank, estradiol levels and genetic data was only available for middle to older aged individuals, thereby not covering the whole lifespan. Furthermore, like our observational studies, data for MR was only available for one time point, not accounting for the time-varying effects of estradiol and other reproductive factors. More generally, other challenges are that the UK Biobank study was not specifically designed with female health research in mind. Although the UK Biobank, for instance, covers the age range for the menopausal transition, the variables related to menopause are quite limited, which restricts our ability to gain detailed insights into how this particular hormonal transition phase affects brain health in females. To address some of these challenges, we strongly believe that coordinated global consortia efforts to harmonize and pool data on female brain health are needed. To this end, we launched in September 2023 the Enigma Neuroendocrinology Working Group. Enigma operates under the motto of crowdsourcing meets neuroscience, and our working group specifically aims at addressing knowledge gaps about the role of sex steroids in the brain across the lifespan. To allow for a lifespan perspective, the working group is composed of 4 subgroups, focusing on puberty, adolescence, menstrual cycle and hormonal contraception, pregnancy and parenthood, and the menopause transition. Even though it's structured into subgroups, the long-term goal of our working group is to pool data covering the whole lifespan to answer pivotal questions about female brain health. If you want to know more about our working group, check out our commentary in Nature Mental Health and our webpage. In case you have relevant data, please reach out and become a part of our working group. In the context of my subgroup on menopause transition, Neda Jahanshad, co-director of Enigma, and I recently received funding from the Welcome Leap Care program to intensify our efforts to study the female brain during midlife using big data and computational methods. This funding translates to 2 postdoc positions, one in the US with Neda and one in Norway, Germany with me. If you're interested, please reach out. Coming to the end of my presentation, I thank you all very much for your attention. I'm regrettably not around to answer your questions, but please reach out to me via email or social media if you would like. Enjoy OHBM.